Okay, so this is the rubric for the grading assignment for the genetic disease project. And I don't know if I can make it bigger. But right here is the box that shows us the, well, this is just terrible. Hold on one second. I'm going to share it with you a different way because that didn't work very well. Give me a second. All right. So now, let's see. Now we want to go to here. Okay, so you can maybe see it a little bit bigger now. Oh, that's not the right paper. I just realized that wasn't what I wanted to pull up. This is it. Okay, so you can see that over here it says the vocabulary notebook booklet, and you have to do each and every one of these things to get full credit for each word. So you have to define the word in your own terms. You have to provide a picture, drawing, or image. You have to provide an example. And then you have to tell me a so what. So tell me why I care that you know this word. Why did I choose this as one of the 50 words that I want to make sure you leave my class knowing? And then the last thing is to answer one of the following, either what is it made of, where does it function, or what process is it part of? So make sure, because when I go through and I grade, I'm looking for those 5 things, and they're all worth points. And if you don't do those 5 things, then I take off points. In terms of the smallest to largest, I know I said indicate relative size using the metric system, and then I have things like ecosystem on there that you can't really measure with a metric system. So in those cases, you can use an example to show why you ordered them the way you did. So for example, you might say that your population's a herd of elephants in wherever, Africa, right? And then your ecosystem would be that herd of elephants plus the grass, right? And then your community would be the herd of elephants plus the grass plus the other things, right? So you can get bigger, right? And you can just use examples in that way. Okay? So but make sure this is what I'm using to grade right here, these where it says instructions. And so those are what you get points for. So make sure you're checking off each one of those for everything. And once you get into a habit, you can just kind of use a template and just fill it in for each one. It should get easier. Any questions on the vocab project? None. All right, and then Ivan's with Amy. Amy's in group 2. Okay, I'm going to go ahead and close out of this. Oh, Gemini, go for it.
Can I still fix my project? Because I didn't know about the so what.
Okay, yes, you can just
Okay.
Resend it to me.
Okay. Thank you.
Yes, I would rather you get it done and actually understand the so what, so that's why. Okay, so that's the first thing I want to share with you. Now, let's look at, let's see if we can pull this up. Oops. So in terms of your genetic disease project, remember, we're only doing this top part, the 1 to 2 page report. In the report, you have to do everything that's bullet pointed here. So you have to describe the disease. Who does it normally affect? What are the symptoms? That kind of thing. Then you have to name the gene and the protein that's encoded by the gene that's involved in this disease. First, tell me the normal function. So if it's an ion transporter, tell me what that does, right? How that affects the cell. If it's a transport protein, right? If it's an extracellular matrix protein, tell me what that protein's normal function does. Then describe to me what's wrong with the gene. So what kind of mutation is it? And then tell me how that mutation affects the protein. Does it make the protein just, no protein gets made, or does a protein just fold improperly, like with sickle cell and hemoglobin? Does it gain some new function? What kind of protein change is it? Then tell me what that protein is doing. Is it functioning in all cells? Is it functioning just in some cells? And then, of course, discuss the genetics, which we haven't gotten to that part yet, but basically you're going to say how it's inherited from one generation to the next. This is the only part you have to do, so all of your points are going to come from this. Basically, right now, this part is not, we're not doing it anymore. I'm not having you do 2 group presentations virtually. It's just too much. Okay? This is where the grades are going to come from. This is what you want to pay attention to. All right. Any questions on the genetic disease project before we move forward? I'll probably go over this again tomorrow in lecture. So if you think of something, we can talk about it then. Okay, so I'm going to stop sharing that one. Oh, no, I'm not. I got to go back. Sorry. All right, then the next thing I want to talk about is I set up a practice exam for exam 4. So make sure you're doing that practice exam to prepare for the test. I do think I am going to push the day of your test back. So if you have any problem with that, let me know, because I can work with you to make it so it fits your schedule better. But I really think I'm going to need Thursday to finish covering material. So I don't think I'm actually going to open the test up until Thursday, and then I'm going to give you Thursday and Friday basically to take the test. And so like I said, if that doesn't work for you, if you can't take it Thursday and Friday because you have something else, like Angela, I know you work, like let's say you work all day Thursday and all day Friday, just let me know and I'll open it up another time when you are available to take it, okay? It'll be like last time, you'll have 2 chances to take it. It won't necessarily be the same questions both times. All right. I haven't built it yet. This one I haven't built yet, so that's something I have to do today is build that test online. So any questions about the test? All right, I got that off my list. All right, now let's talk about the research paper. Let's see, where did I put that? Okay, so most of you have submitted to me your papers for your research paper. What I'm finding is that I'm not sure you're paying attention to this rubric. So if you can't find this rubric, email me or something, and I will email it to you so that you have it on hand. But what I'm finding is that the additional things that I've asked you to do for this paper aren't being done properly. So you have your original introduction, and then you have to make sure you have your big picture part of it put in. So that's these numbers right here. You have to talk about the background on your experimental question. That's your big picture. Relate it to your project. Tell me what you plan to do. Generally summarize what you're going to do. So a lot of people are missing these 12 points right here, getting very few of those 12 points. In terms of methods, a lot of you are still missing these first methods. So don't forget, you have to have a method that says characterization of Saccharomyces cerevisiae or whatever your organism is, and then you say, I stained it with methylene blue, looked at it under a microscope, and measured it using a calibrated microscope. Make sure you're saying those things. Most of you did really good on this part because you had already turned this part into me, so that was really fabulous. For your results, you have to have written results for both your characterization of your organism and your environmental experiment. And what that means is you have to have a paragraph that says, this is what we did, and we found that the cells were gram stained to a purple color, they were bacillus shaped, and they were 2 microns in size. And then you refer to figure 1 to show me a picture of that. And then your figure 1 is a picture of your organism with a caption. So these 3 points right here, written description, that's your paragraph. Critical values emphasize that you have to at least tell me cell size and shape. If there's a color or a gram stain, you can use that as well. Then 5 points goes to just having the image of your organism and then a caption that tells me magnification and about your picture. All right, so make sure you're doing that. Some of you are still missing this part right here. And then you have to have a results section for your experiment. Same thing, you have to have a paragraph that actually states your results. So you're going to say, to understand the effects of disinfectants on yeast, a zone of inhibition assay was done. Say it louder. I thought I heard somebody. Okay, a zone of inhibition assay was done. Our control was bleach, and the zone was typically this, on average, this size. Comparatively, the zones around the lemon juice and the vinegar were this many centimeters. You're not analyzing it, you're just telling me the numbers. All right, and then you'll say, see figure 2, which is going to be a graph of that data. So you have to mention your graphs in the paper. All figures are mentioned. You don't include your tables here. Instead, your tables should be presented in the appendix. And you all have nice tables because I gave it to you, so make sure you do that. So have tables for each experiment that should have a caption, and then it should be presented in the appendix. And then, of course, your graphs, which your graphs look pretty good, should be part of the paper. So you have a paragraph about your data, then your graph. Some of you had 2 kinds of data, so you'll have another paragraph and then another graph for your second kind of data. Okay? And then the last part, you're going to summarize what you did for the entire project. So first you're going to say, to better understand the cells, they were stained and viewed under the microscope and measured. They came out to be gram-positive bacillus-shaped. This confirms that they are prokaryote and E. coli, right? That's all you really have to say for that part. And then you're going to summarize your conclusions for the environmental experiment. You're going to say, to better understand how essential oils affect the growth of bacteria, they were grown in these different environments. The control was this. This is what we found. Comparatively, we found this, maybe oregano oil, didn't have a zone of inhibition at all, indicating that oregano oil is likely not a good antimicrobial. So this is where you get to say, whatever you tested, was that something that supported your hypothesis or not, right? So make sure you're relating what you found back to the hypothesis, back to the big picture question. Okay, for this next section here, we didn't really have problems encountered because we didn't do the experiment, so that's probably not something you have to worry about, but you can say future experiments. So based on what you found, what would be your next question that you would want to ask? All right, make sure when you relate it back to the big picture or to any other sort of scientific principle, that you use citations to support that information. So I'm looking for citations in the discussion conclusions. I'm looking for citations in the introduction. Those are the 2 places. If you don't have citations in the methods, I don't take off points, but I do if you don't have it in the introduction or the conclusions. Okay? Keep in mind some things I'm still seeing that need fixing is on your graphs, the title of your graph should be in sentence case. That means every word should be capitalized. You always put the name of your organism in italics, even in your graphs, right? So that's important to do that as well. Do a spell check. Make sure you're, as much as possible, using good grammar, right? I don't necessarily, it has to be pretty bad for me to take off for grammar, but if it's pretty bad, I will take off for grammar, okay? So you still get one more chance to make this right. So if you've already turned this into me and I already gave you your grade, don't worry about it, but please take it seriously to make sure you're fulfilling this rubric. Grade yourself. Did you do these things? Okay? Any questions on that? No? We're good? Okay, so then the next thing I want to talk about, I know one more thing. So once I grade your paper, I will fill this form out and send it to you via email with comments over here and your grade assignment here in these numbers. I also make comments in Turnitin. So let me see if I can pull up someone's Turnitin. I think I have one right here. Okay. So when you go in, you'll see, oh, you can't see this. Hold on. I got to put on this screen. Can you see it now? Okay. So this is not one I've graded yet, so you won't see anything. So this is Ashley's paper. But when you go in and look at your paper, if you click on this, like it looks like a piece of paper with a pencil in it, when you click there, you'll see where I have written comments. So comments from me will be right here. And then if you look within the paper and you see any sort of little bubble out like this, when you click on it, I might add a particular comment for that one section of your paper. So maybe I don't think you need this word, and so I say this is a better word or something like that. So there's basically 3 places you have to go look for all the comments. You have to look at the rubric comments that I send you in an email, so it'll be on this. Then you have to go into the grade book, go to your paper. So there's a little, I can even show you. So next to, here I'll go to some other class so you don't have to see your own. When you go into your grade book, these people don't have me for labs, so don't look at their papers. Let's see. Here, when you go into the grade book, right next to your grade, right? So you can click on that and then hit attempt. And what that will do is that will allow you to see it. And when you pull it up, then you can actually see the comments that you were given and Turnitin. So all those little bubbles will pop up. If you click on this, any of the text I wrote there will pop up. Does that make sense? So make sure when you're doing your final, because you only get one more chance now. That's it, just one more time, okay? So this time you really, 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 really need to pay attention to the feedback. If you end up liking your grade over the draft you just did, you don't have to do the revisions. You can opt out. Like you can just say, hey, I am happy with an 89. I want to leave it there. I've got too much other stuff to focus on. That's fine, right? But if you got anything under an 80, I would say you want to spend the time doing the revisions. Any questions? Okay, so now what I'd like to do, and I don't know how many groups I have here. Let's see who came in. So Irwa came. Hi, Irwa. And Alexis and Camelia. So Irwa, you're in the same group with Gemini, correct? That's group 4.
Yeah.
And so is Camelia. So you guys are going to be in your group. And Alexis, you're in the same group as Amy and, is that right, Amy and Ivan?
No, Ashley is the one.
Oh, Ashley. I always get Ashley and Alexis mixed up. Is that not the craziest thing? So Alexis, you're in the group with?
I'm only. Karen's not in here unless she dropped.
Okay. I don't think she dropped, but Ramon, you were with Ramon? No.
No,
Ramon?
I was with Bree and Karen.
Who was the other person?
Bree and Karen.
Okay. All right. So I have Elaine's group, Elaine, Grace, and Gretchen. I'm going to break you out in your groups, and what I want you to do is, if you haven't already, use the template. Oh, I didn't show you the template. I'm going to show you in just a second. Use the template to figure out who's going to contribute what to the poster and start to build it. You can even be doing it remotely if you want to. So let me show you this. All right, so this is the template that you can access from online. All right. These are not the words you leave on your poster, right? You write your own words. So you're going to replace describe the work here with your title, right? And I've been giving you feedback on your title and your paper, so that should be a helpful thing. Then you're going to put your name, your group's name, everything here, okay? Between the 3 or 2 or however many of you there are, you're going to figure out which one of you think wrote the best abstract. And that might change once I start getting papers back, but for right now, it doesn't really matter. Choose who you think wrote the best abstract. And the abstract can literally just be copy-pasted into this section. So there's not a lot of work to be done on the abstract part. With your introduction, you're not going to just insert your introduction here. It's going to need a little bit of work. You need to focus on what's my big picture question and some background on my organism, and then relate your organism to the big picture question to kind of prepare us for what's to come. All right. Your methods can just be that little short paragraph you guys wrote for me, plus you looked at it through the microscope. So that's totally fine. Okay, in this middle section, I'm going to show you some examples in a second. In this middle section, you're going to put your results. So this is going to be, you know, maybe characterization of Saccharomyces cerevisiae, and then you're going to put your picture of your organism here with a caption. Keep in mind, I just told you that in your paper, you had to have a paragraph that told me your results, and then you had to have an image that had a caption that told me your results. On your paper, on your poster, you don't need the paragraph. You just need the image and a good, well-written caption. Same goes with your environmental experiment. You are going to put your graphs with very well-written captions with them, and that's all you need. All right, then someone else will get assigned this part of your poster, right? In the discussion, you're going to highlight your major findings and what they mean. Talk about how it relates to the big picture question that you were asking. Maybe talk about what you would do differently if you had an opportunity to do it again. Then you're going to talk about literature cited, and then any sort of acknowledgments. And I think last week Hannah put out the names of who you should be acknowledging. Okay, so let's look at some examples. Here's one from a couple semesters ago. Here's their abstract. Notice what they did in their introduction was they, instead of making it a paragraph and very wordy, they bullet pointed it. So they said, okay, fertilizer runoff is a problem. Oh, big picture, right? What's the problem? Algae blooms can be a result of them and hurt organisms. So this study addresses a particular algae and what we can do to maybe stop these blooms, something like that, right? So you see how they kind of went from big picture down to smaller picture. You can see here they kind of arranged a little differently. They didn't have a lot of space down here, so they put their acknowledgments here and moved their methods over there. Totally fine to do that. You can arrange it however you want. All right, then they did their results. They have their image here, and then here's their caption. And then they have their graphs here, and then here's their graphs captions. Now, some feedback about this one is I'd say the images are pretty good. I'm not sure you needed 2. You probably could have spaced things out and made things bigger with only 1, because I feel like the graphs are pretty small and hard to see. Here's their materials and methods, and then here's your discussion. Based on our study, our hypothesis was supported that increasing nitrogen concentrations increase the growth of Anabaena, blah, blah, blah. Future studies, right? And then there's your literature cited. So do you kind of see how this should be organized? Any questions on that? Amy?
Yes, so it needs to be in this form. So our group was like create a PowerPoint about it, but
Okay.
as you show us right now, we have to put it in this like.
I would prefer it in this form because this is what the poster would look like. If you really struggle to get it in this form and you just want to do a PowerPoint, I suppose that's okay, but I would prefer it not to be.
Oh, okay, sure. Thank you.
Uh-huh. So let me see something real quick. I thought I had, it was, let me see if this one is it. So Amy, can you see this one? This was a group that they couldn't figure out how to figure out that template. So they did each section as a separate slide. See how that works?
Yes, this is how our group did
Right.
our.
So see if you can get it into that other format, but if you can't, this is acceptable as well.
Okay, sure. Thank you.
All right. Let me show you another one, just because it's nice to see different ways it can be put together, just to show you that you can be creative with how you want to do it. This one, here's their title. They got kind of creative and made like a funky background. They were working with Chlorella, so they had a picture that they used that they put in the background there. They have their abstract. Notice their abstract. This one starts broad with the big picture and gets more narrow. So the prevalence of diabetes is on the rise. Chlorella has been said to contain sugar-reducing properties. This study was carried out to determine if these claims are true. Oh, so now I totally know what this is going to be about, right? And then in their introduction, here's what Chlorella is. Here's how we're relating it to the big picture. Here's what we did. We asked, these people did in questions, can Chlorella really be beneficial to diabetic patients? This study addresses these questions. And then they have their methods. Now their picture is a little bit smaller. It's kind of the opposite problem of the last poster. Their picture here is really small, but their graphs are nice and big. I really like their graphs. I could see very clearly what they were doing. Shut that door, please, buddy. I can see very clearly that they were looking at glucose with sunlight and glucose without sunlight to see if sunlight made a difference in the growth of their organism. All right, and then over here they discussed what their findings meant. I think I have one more to show you. I thought I did. Let me go back here. Poster. Was it this one? Nope, I showed you that one. Poster preview. Okay, here's another one. All right, so this was another one they kind of did a little bit differently. They couldn't figure out the PowerPoint template, so they just did it in Word. They figured out how to kind of just paste it all in and make their own document in Word, and maybe you can do it that way, that's fine. But they did their abstract, their intro. You can see their cell size here. There are lots of graphs, and then there are other things. So you guys kind of seeing how I want you to put this together?
Yes.
Okay.
Yes.
So I'm going to put you in your groups, and I'll pop in and Hannah will pop in. Do you all have the template available? I got to make everybody, hold on one second. I got to make you all presenters so that you can share with each other. So if one of you in every group could pull up the template, I'm pretty sure you can share that with your group once we get there. We'll see once we get there. So I wish I could do this all at once, but it doesn't look like I can. And Hannah, I'm going to make you a moderator.
Okay, sounds good.
Where's Hannah Haas? There she is. Okay. All right, here we go. We're going to go in our groups, and then Hannah and I will pop into your groups to offer a guidance. Mohammed, remind me who's in your group. Angela, right?
Yes, Angel.
Okay. I'm going to put you in Angela's group, and then Alexis, you're by yourself, right?
Yes.
So I'm going to put you in your own group, and you can just kind of work on it by yourself until we get there with you, okay? All right,
Okay.
so here comes the groups, and I hope you can all talk. Let's see if it works. Let me see what this looks like. Okay. And start. All right.